Hello everybody, this is a recording made after the class because there are a couple of problems with the recording made in the class, which is why the videos for this lecture are separated into several parts. Okay, so in the last class, in class 2, we investigated how the computations performed by neurons in the brain represent and process information, and how these computations provide the inspiration for operations performed in deep convolutional neural networks, artificial deep convolutional neural networks, that is. The principles involved here apply equally well anywhere in the brain. The organization of neurons is very similar wherever you look in the brain. But when we start looking at the results and implications of this processing, it becomes very hard to talk in general terms, and it becomes very useful to follow through a specific example, which we're going to do for early visual processing today. So we used early visual the early human visual system as an example in the last class and also today for a couple of reasons. First of all, computer vision problems like object recognition are exceptionally difficult to solve using formal rules, and so they've been an excellent application for artificial deep networks, okay, particularly in the case of object recognition. The second good reason to study this is that the early visual system is probably the best understood system in the human brain. Here we really understand at the computational level how the brain is working things out, how features are being extracted. All right. And this is largely because it's very easy to precisely control visual inputs. We can use a computer screen to do that these days, right? And secondly, it's because we have very good animal models of the visual system, particularly an animal called the rhesus macaque, which is a monkey that we'll see come up a few times in this class. However, as I mentioned before, we believe that very similar principles operate in other networks in the brain. Okay. So we saw in the last class how the retina and the start of the primary visual cortex separate images into multiple feature maps for different colors, for different edge orientations, also for the two eyes, and we're gonna look today also about motion as well. So these features can be derived using relatively simple filters operating over space and time and different feature maps. Okay. So today we're going to look at how some of that works, and we're going to look at subsequent stages of visual processing where the filters become a little bit harder to characterize and understand and may indeed result from learning, so rather than being hardwired. Okay. So on leaving the retina, the ganglion cells that we looked at in the retina last time feed into the visual cortex, the primary visual cortex, which you see at the back of the brain here, via the thalamus's lateral geniculate nucleus. The lateral geniculate nucleus, or LGN, is effectively just a relay station, and it doesn't really perform any transformations of the image. So we're going to mostly ignore that. It's going to come up a couple of times, but it doesn't really do much, so we're not really going to focus on that. So during this process, the information from the two eyes is joined together. Okay, so if we look in the left hemisphere of the primary visual cortex, this is responding to the left half of both of the two retinas. Okay, that is to say, the right half of the retinal image. Okay, so here both of the two eyes images from the left side of the retina on the two eyes is joined together, and the other half of the image is passed to the right hemisphere, to the other hemisphere. So, so one thing that we discussed briefly in the last class is that when we look at the cortical surface, the neural representation here is also laid out as a map of visual position following the layout of the retina. So sites, the result of this is that sites responding to particular image positions are held together. Okay. So this was first determined by showing a busy sort of dartboard pattern like this with flashing lights in the little black and white lines that you see on this image here, and then doing this to a monkey where you're also staining the cortical surface of the primary visual cortex for metabolic activity, so that the locations on the cortical surface that are activated by this pattern become dark on the cortical surface. And you can see here an image of the primary visual cortex of a monkey that's had that done. And what you see here is that the dartboard grid that we're seeing here is kind of mirrored onto the cortical surface. So the neuronal representation on the cortical surface is following that structure that we're seeing from the retina. Okay. So as a result, the important thing to remember here is that spatial relationships from the image become spatial relationships in the neural activity of the cortex. Right? And this allows filtering stages, subsequent filtering stages, to have a limited cortical extent and a limited spatial extent also in the feature map. So that's going to become a bit clearer later. So then this allows interactions over minimal distances, right? Something we mentioned briefly last time is that cells that wire together, cells that form strong connections together, normally because they are responding to similar things, also tend to lie together in the cortex for efficient interactions. However, there is some separation here between the representations of the features carried by the parasol cells, the midget cells, and the bistratified cells, which are different cell groups carrying information up from the retina. We mentioned briefly last time that the parasol cells are cells with relatively large receptive fields that respond to very quick features in the image, so that they respond under low light conditions and they respond very quickly, so they give good information for subsequently computing motion in the image. Right? The midget cells, on the other hand, have smaller receptive fields, so they give finer spatial detail, and they also have color selective responses. Right? They're carrying color information and very, very fine spatial information as well. The bistratified cells only really carry color information, and we're not going to look at those too much. Okay. So when these feed forward into the primary visual cortex, they end up in slightly different places. Okay. All of the visual information is coming into the visual cortex in this retinotopic map organization, but there's a slight separation between the color information and the sort of information that's better for detecting motion. These different cell groups are held in slightly different depths or layers of the grey matter. It's important to point out here that grey matter layers are not equivalent to neural network layers, but here we do see several neural network layer-like stages. Okay, so that is to say, we see different numbers of synapse steps away from the photoreceptor input as we move from left to right across this chart in the top of the slide. Different layers of a neural network are sometimes held as different layers of grey matter in the same cortical area, but more often held as different cortical areas. So there's some spatial relationship between the cell populations from the retina here, but they're also held in the same visual area at this stage. Okay, so spatial relationships allow neurons to interact most easily if they're carrying information about the same feature type, as these will be closest together. But those four other feature types are still nearby, allowing the brain to easily analyze relationships between different stimulus characteristics found in the same location by using filters that span all of these different feature maps, so have some extent over the different separated layers that we're looking at here. Okay. Responses to specific orientations, that is to say specific directions of lines, first emerge in the primary visual cortex. In the primary visual cortex, we start finding cells that respond when an edge has a specific orientation, which we call the preferred orientation, okay, and is shown also in a specific position. It remains very important for these cells that the stimulus is shown in a particular position, as well as at the right orientation. Now, Orientation selectivity, like we see here, can be, so let's just talk through this diagram for a minute, right? So here we can see that if we show bar A, which is in the middle in the on region of this cell, that will cause the cell to become active. So we see a higher firing rate in the middle of this trace at the top here. If we show if we show the same orientation in a slightly different position, in position B, right, then the cell will be inhibited because the bar is falling in the inhibitory surround. If we show it in orientation C, where it's in the right position, at least the center of the bar is in the same position as in A, the cell isn't going to change its response at all because we're stimulating a similar amount of the on region, the sort of excitatory region of the receptive field, and also of the inhibitory regions of the receptive field. Okay. Now, orientation selectivity like this can be built up from spatial interactions between different center surround retinal ganglion cells, like we see on the left here. Okay. So these retinal ganglion cells, which here they're shown as lateral geniculate nucleus cells, which are in the thalamus, as I say, these are very, very similar types of cells. Here, these three cells, if all of these are active, okay, that means that we have a vertical bar running right down the middle of them through all of those three on regions that we see on the left here. Okay, so that will basically produce a response like we see in the middle here, that a bar at position A will produce a strong response, but if it's at position B, it's going to be falling in the off regions of all of those lateral geniculate nucleus cells, and if it's in position C, it's going to hit some of the on regions and some of the off regions. Okay. So expressed as a convolutional filter, it might look something like this, all right? But remember that the inputs to this filter have already had a suppressive surround organization. So again, it can be very misleading to think of this in terms of an image. It's much more useful to think of it as a pattern of neural activity than an image of the bar. But nevertheless, if we show this convolutional filter, a light bar at position A running right down the middle of this convolutional filter is going to hit all of these 2s and none of the -1s. All right, so that's going to produce a strong positive response like we see in the top trace. If we show bar B, it's going to fall in the -1 areas, right, only. All right, so this is going to inhibit the cell. It's going to set its activity below baseline. Okay, and if we show bar C, which is running diagonally across this filter, it will sort of stimulate the 2 in the middle, but it will also stimulate the 2 -1s on the side, so they'll have no change. All right. This is a way of thinking of how this might be implemented in an artificial deep convolutional neural network. Okay, so as I said, it's very important to try to avoid thinking of this like an image, the input as like an image, because the input to this process comes from the output of an earlier process, all right? So it's a pattern of neural activity. Humans are really bad at thinking like this in terms of transformations of representations rather than transformations of images. Conveniently, neurons and computers don't have this limitation. They transform whatever input they're given. But at this early stage, we can easily make an image that gives an idea of where the contrast in the image is, okay, and so gives an idea of the pattern of outputs that the retinal ganglion cells might have. Okay, so this is the image on the right here. So where we have a really, really early representation, we can think that, well, the image doesn't look like the image anymore. It looks like a pattern of neural activity that we see in this image on the right instead. Okay, so we have in the primary visual cortex, we have orientation selective cells. Okay, at the coarse scale, neurons processing the same part of the visual field are grouped together. Now, neurons with similar orientation preferences are also grouped together, but this grouping is at a much finer scale, kind of within the subscale within the same position on the cortex. Okay, so over a very, very short distance, here just a couple of millimeters, we see a progression through all of the different orientations, all the different possible orientation preferences. So this very fine scale is called the scale of a cortical column, okay, and each column contains neurons responding to the same position but with different preferences for orientation. So these different orientation columns effectively form further feature maps. However, I will mention at this stage that if we were, sorry, I'll come back to this point actually. Now, so basically, if we get an electrode and we slowly pass it along the cortical surface, as we see in this inset image in pink at the top here, then we're going to slowly hit a progressive range of different cells, and all of these have slightly different orientation preferences, gradually changing along this electrode track. It's since been shown using two-dimensional optical imaging techniques that orientation preferences change in 2 dimensions across the cortex. Okay. This description of, if you look at these, here we've got the orientation preferences color-coded. If we look at these, we can kind of follow these kind of rainbow progressions of different orientation preferences gradually changing across the cortex. We see little stars here, which are showing where we kind of have a discontinuity where all of the nearby orientation preferences can be very different, basically because there's no way to kind of fit circular spaces into a two-dimensional surface without having kind of points where all of these preferences come together. So this description of the orientation organization is referred to as orientation pinwheel. Okay. This description of two-dimensional orientation pinwheels has superseded the description of columns that is apparent when advancing an electrode along a one-dimensional line, because we can see the two-dimensional surface of the cortex here. Okay. Of course, if you were to take a little part of this, this map and you made a one-dimensional line along it, we would see a gradual progression of orientation preferences along that line, like we saw in the last image. So, we've seen this separation into feature maps for different orientations before in the context of artificial deep convolutional neural networks. Here we can see an input image which passes through filters with different orientations, producing several different feature maps representing the amount of image content at each of these orientations. So again, at this level, at this orientation column or pinwheel level, neurons that have similar response preferences are found together. This can be very computationally useful because if we have a filter that will accept a range of nearby orientations, then a neuron implementing this filter can synapse with a group of nearby neurons. Okay, so if we have a particular range of orientations that all are basically equivalent or are all very similar, okay, then we can use a filter that just has a very, very small size in this cortical column to synapse with all of those. Okay. In a biological network, it's worth pointing out that these orientations can have any value. Okay. In the artificial network that we saw before, here we're using 8 different filters, and this produces 8 different feature maps. Well, in a biological network, we can have any value of orientation. We're not limited to a fixed number of discrete filters and feature maps. Instead, it's more like orientation is an entire continuous third dimension of this feature space, with position forming 2 of the dimensions and orientation forming the third. So, neighbors within this third dimension, okay, in this orientation dimension, also have similar responses, and therefore filters can have a meaningful extent in this third dimension. Okay, that is to say that a filter might only sample from a very narrow range of orientations. Okay, so wouldn't necessarily touch any of the other orientations that it's not interested in at all. Okay. So this is not the case in artificial deep networks, where filters typically span all of the feature maps in a layer and therefore can learn any relationship between different feature maps. However, if analysing relationships between nearby feature maps is the most useful for task performance in an artificial network, then filters can develop that have no weight, have like a 0 weight for further away feature maps, okay, which will have the effect of focusing on relationships between nearer feature maps. Constraining an artificial network filter to a neighbouring range of feature maps is rarely done, though it's certainly possible and may have some computational advantages. It's not normally done. Okay. The grey matter in the brain is arranged as a folded 2-dimensional sheet. and it doesn't have access to a third spatial dimension to put orientation in. Okay. However, in artificial deep convolutional networks, filters can effectively span a third dimension across multiple feature maps at each network level. Somehow, the brain creates an organized mixture of these different orientation components, okay, into this, kind of fitting all of these different orientation preferences down, squeezing it into one sheet like this, in one two-dimensional sheet of cortex. Okay. So how do we go from this kind of third dimension to kind of forcing that third dimension into the existing two dimensions of the cortex to give this intermixed representation of different orientations? Super recent work, just from this year, and has not even yet been published, has simulated how this might happen in an artificial deep convolutional neural network. Okay. Here, we see two spatial dimensions in x and y here, and the third dimension, which is shown as a stack in different colors, is giving us different orientation filters. Okay. Different feature maps for different orientations, pardon me. Okay. So, instead of using a deep convolutional neural network with independent channels like this, multiple channels, all of the units in the first layer of this network have been forced into the same two-dimensional feature map. Okay, so here they've all been placed onto a single two-dimensional surface and randomly placed initially. Okay. So a convolutional filter leading to the next layer must sample within this two-dimensional feature map that we see here, where all of those different orientations have been put together. As part of the machine learning process, so these filters might look something like this. So these are the same filters, right? The same orientations in different colors here at different positions within the map. Okay. As part of the machine learning process, units within the same x and y position, so within each of these smaller squares here, okay, can be moved around, and the rule is such that they will tend to get closer if they respond more similarly, or they respond at the same time. So basically they're forcing into this network the rule that if things are wiring together and firing together, they're also brought to lie together as well. Okay. When they do this, if we look at where those units end up, units with similar orientation preferences, which is, say, the individual pixels in this image, come together, and they self-organize into structures that closely resemble orientation pinwheels. So, by including a spatial constraint in the learning rules, it becomes clear that neurons with similar responses, which will tend to respond together, will also tend to lie together. So these are kind of two sides of the same coin, right? That we've got neurons that respond together will tend to form stronger connections, and neurons that form stronger connections, therefore, will tend to be closer together because that's a more efficient thing to do. Okay. This study also found that the same organization did not emerge if the same filter was used at all spatial positions. That is to say, if weights were shared across the layer. Okay. So we saw in the previous classes that in artificial deep convolutional neural networks, weights are almost always shared across a whole layer. This is here they implement a network where the weights aren't shared, where a different filter can develop for every different spatial position in the image. Okay. If they used shared weights, what they found was a homogeneous mixture of orientation preferences emerging. Okay. So that is to say that rather than this happening, right, that you get a clear separation of different orientation preferences and gradual progressions in them, okay, instead you just get just a completely random mix of these different colors. Okay. So therefore it means that basically this can happen in a biological network, but only because the weights are not shared across different visual positions. Okay. So what they had to implement here was independent weights at every filter location. These filters then could differ across the image, and indeed they did differ across the image in some interesting ways. If we look at the top right group of filters that we see here, these all have like not much structure to them, right? So there aren't sort of strong edges within these filters. They tend to be pretty homogeneous regions. That is the filter that emerges in the here, the top left corner of the image. Okay, that's the set of filters that emerges for the top left corner of the image. And these images are photographs that humans have taken, and humans have pointed the camera at the thing that they think is interesting, right? And therefore, the thing in the top corner of the image is probably nothing in particular. It's probably just some background without much feature to it. Okay. If we look in the center of the image, we see finer scale features. All right, so this is what we see in these bottom two rows on the right here. We see very fine scale features with lots of structure to them. So if we look at what happens with shared weights, though, we have to have the same. This is you see on the left here. You have to have the same filters for all of the different image positions, and these can't vary across the image at all because the weights have to be shared. Now, this is a very, very computationally expensive thing to do, as a new set of weights must be learned for each of 54 by 54 image positions in this particular example. Each of these filters, has a large number of weights within it. So it has a large number of parameters that must be learned. Okay, so each of these filters has 11 by 11 pixels crossing 3 different color channels. Okay, if you have to learn 96 possible filters, which is what they use in this case, right, in total, and each of those filters has 11 by 11 by 3 weights. Okay. That means that you end up with about 35,000 parameters that are targets for machine learning that have to be learned. However, if, as you see on the right here, you also have 96 channels at every position, and each of those channels contains an 11 by 11 by 3 filter, and there are 54 by 54 positions. That means you go from 35,000 parameters to 100 million parameters. Right, so that's a very, very large increase in the computational complexity of the machine learning. As we discussed last time, this is not a problem for biological neurons, only for their artificial simulations. Okay, so to summarize what we've seen about orientation selectivity, we've seen that contrast is initially computed in the retinal ganglion cell using an orientation independent filter, so just a filter that responds best to a point of light surrounded by darkness. Artificial deep convolutional neural networks actually avoid this step, going directly from the image to orientation-based representation. Okay, why that doesn't happen in the human visual system is probably because detecting contrast basically only picks up where the image is changing. All right, so it's basically throwing away a lot of the information. Okay, and that allows this kind of compressed form of information to be sent down the optic nerve, which is quite a thin nerve, up to the brain. All right. If you had orientation filtering happening before that, you're turning each of the filters that we see for a point of light into multiple filters for different orientations. So basically you're expanding the information back out again. Right? So it may be that we have this sort of retinal ganglion cell stage where we're just detecting where there's points of light, or we're having responses to in an orientation independent way to contrast. Right? That might be useful simply because it compresses the information to send it down the optic nerve, which of course is not a constraint for artificial deep convolutional neural networks. Okay. We've also seen then that orientation selective responses are computed in the primary visual cortex, V1, by operations that compare the outputs of retinal ganglion cells. We've seen that orientation preferences gradually change across the cortex, but at a much finer scale than the spatial visual field maps that we've seen. And what this does is it squeezes multiple feature maps into the same two-dimensional cortical surface. This may arise to optimize connection lengths. All right? And indeed, if you make a simulation that is aiming to optimize connection lengths, this is what happens. This emerges spontaneously. But this only happens when filters are not shared across the network layer's image representation, because that basically forces all of the orientation preferences to be homogeneous across the image. So we see a homogeneous mixture of orientation preferences. Okay. It's fairly, so we've looked at orientation a lot. We're going to quickly look at a couple of other features that are important for the early visual system. So the first of these is motion in the image, right? So it's fairly rare for artificial deep networks to consider changes happening over time. Generally, artificial deep networks are given single static images as inputs rather than movies, largely because of computational limitations. But of course, biological visual systems inherently receive inputs that change over time. So, biological systems have a mechanism to detect motion and to detect its speed and direction. Here we see a neuron with, well, actually it's a circuit of 3 neurons with 2 spatially separated inputs, and one of these has a delay on it. So here we can see that if this dot that we see at the top here is moving from the left of this image to the right, it's going to hit the neuron on the left's receptive field first, and then it's going to hit the neuron on the right's receptive field slightly later. Now, the neuron at X here is only going to respond if it receives inputs from both of these earlier neurons at the same time. Okay, so that is to say it's going to receive excitatory postsynaptic potentials from both of these neurons above it, but they'll only reach a threshold if they arrive at the same time. So, in order to detect the direction of motion, we insert a delay into the conduction of one of the neurons. So one of these neurons is going to take a little while to hold the response before passing it on. So then the signals will reach point X simultaneously, only if the length of that delay corresponds to the time it takes the moving dot at the top to cross the span between the 2 receptive fields. Okay. So this is what this looks like as a kind of video. Okay. So here we see the point of light hitting both of these neurons, one of them being delayed a bit by that cylinder there, and then they both rise at the 3rd neuron at the bottom at the same time, so that fires, and that detects motion with a particular speed and a particular orientation. This can be expressed as a filter with a time dimension and, for simplicity, only one spatial dimension. Right? Here the activation threshold would be 1, so that if the stimulus activates position 1 at time 1 and activates position 2 at time 2, then we will reach that threshold and the filter will lead to activity. Okay. This filter is specific for one speed. That is to say, the ratio, that speed is the ratio between the span and the delay. Okay, so that is to say a certain number of degrees of retina per second of delay. We can therefore change the speed that makes this response by changing the delay. If we increase the delay, okay, so in this case we're going to double the delay to 2 delta, right? Then this will give a response when we show a slower speed. Okay, so if it takes the same time, a longer time to cross the same span, all right, that will produce the response. Okay, so again, we can express that as a convolutional filter, where we'll see a response if we have position 1, that is, say, the position on the left, stimulated at time 1, and position 2 stimulated at time 3. All right, so then it gives kind of 2 units of time between it, so it will respond to the same change in distance for a greater change in time, so that is, say, a slower speed. Okay. And we can also make a circuit that will respond to a faster speed in the same direction by increasing the span. So here we've doubled the span, but we've kept the same delay as we had in the first case. So if in the same amount of time our stimulus is travelling twice the distance, then it's travelling at a faster speed. This can also be expressed as a convolutional filter in terms of both time and space. So here we see that if we hit position 1 at time 1, and we hit position 3, so after a further distance at time 2, then we'll see a response to a faster speed. So now we've seen how the brain can compute responses to motion as well as orientation, and we looked in the last class about responses to colour as well. All of these are important features for us to be able to understand. So I'm about to show you a little video that, that kind of shows how the visual image is separated out into these different components using a little simulation.
This TV picture gives you a good impression of depths and surfaces. The information in the picture is defined by brightness and color and how they change over time. What would this picture look like if you could only see in color? Color information lets you see the surfaces. But the fine spatial detail and impression of depth is certainly reduced, maybe even lost. This picture, defined only by brightness, gives fine spatial detail. However, in order to obtain the detail, the human visual system gives up temporal resolution. And this is what it looks like. It's difficult, in this slowly changing image, to connect the sound of my voice to the motions of my mouth. What would this picture look like if all you could see were the parts that were moving? Something like this. Now only moving edges in the image are visible. If an object stops moving, it disappears. The original image we looked at was a combination of these three attributes: brightness, color, and motion.
So we can see there that just what we've seen there is the image split up in separate components, which is more or less what the visual system is doing, even though we perceive all of those as happening together. Okay. The primary visual cortex also receives separate inputs from the two eyes, which kind of doubles up on all of these feature maps. These are initially kept separate as distinct cortical columns for the left eye and the right eye. Various methods can mark the parts of V1 receiving inputs from one eye. For example, we can again stain V1 for metabolic activity while covering one eye of an animal and showing it images in the other eye. Okay. And this shows that there's a distinct pattern of interlaced columns or feature maps for the two eyes. These feed into neurons which integrate the responses from the two eyes, effectively with separate spatial receptive fields for the two eyes. And this means that cells respond to a particular position difference between the two eyes. If we look at these images here, the cross is where we're looking. If we look at relative to the cross, the blue rectangle is in the same position in the two images, right? While the black and red rectangles are in different positions in the two images. They start on the left of the image in the left eye, and they're on the right of the image in the right eye. Okay. So if we then compare what is different between these two different receptive fields, between these two different images that the two eyes are receiving, then we can work out how far away something is, because different distances produce different projections on the retina relative to where we're looking in depth. Okay. So this means that these cells, which have two spatially separated receptive fields in the two eyes, respond to a particular position difference between the two eyes, which reflects the distance of the object from the viewer's fixation in depth. This underlies a large part of human depth perception. So, if we look at the responses of V1, primary visual cortex, we see responses to the individual images in the two eyes, right? But after V1, here shown in V2, we see responses that reflect the integrated perception from both of the eyes. Okay, so these different elements effectively form several distinct feature maps that are all mixed together at the same cortical location for the same image location. A full set of feature maps contains responses to both of the two eyes, right? Also contains separate responses to color in these blob areas that we see here that kind of specialize in color processing, and also all of the different possible responses to all of the different possible orientations in an orientation pinwheel. Okay. A full set of feature maps for a single image location is called a hypercolumn, and this takes up about 2 by 2 millimeters of the cortical surface. That's a number that's going to come up later, but basically 2 by 2 millimeters of the cortical surface holds basically all of the information, all of the feature maps that we could have about a single image location. Okay, so far we've seen that in V1 there are a relatively large and complex set of feature maps with each feature represented at all spatial positions. As a result, V1 is a very large brain area because it has to have all of this information together. Okay. This set of multiple feature representations feeds into subsequent processing stages, and it's important to remember what these different feature maps in V1 are. So first of all, we've got the different color feature maps for red, green, blue, yellow, light and dark, which we saw in the last class. We have separate maps for the two eyes. We have multiple orientation representations, and indeed it's a continuum of orientations rather than being sort of a discrete sampling of orientations. Similarly, we have representation of different motion directions, okay, by basically having spatial offsets between the two receptive fields in the motion detector in different directions, and also different speeds. And again, both of these are continuous, unlike in artificial deep convolutional neural networks. So this gives a really rich set of information about the input image that has been broken down a little to extract some really, really important features, right, like color, depth, orientation and motion that form very, very useful inputs to subsequent stages of processing.
So this large and complex set of feature maps that we see in the primary visual cortex forms the input to subsequent processing stages. Okay. In V1, different features are processed in intermixed columns within the hypercolumn, and something very similar happens in the next areas, V2 and V3. After V3, though, form and motion processing are often processed separately and in different areas. Okay. One of these pathways specializes in motion and space perception, which is this pathway going up into the parietal lobe, known as the dorsal pathway. Okay. While the other is mainly involved in object recognition, which is the pathway we see down here towards the inferior temporal cortex. These are effectively two deep neural networks processing information relatively independently but simultaneously to achieve different tasks. That is to say, recognizing what an object is, recognizing where it is and how we might interact with it. The network for object recognition has been simulated in artificial networks very extensively. We'll look at that a lot in the next class. The network for motion and space processing is not. And it's relatively part of the reason why it's not particularly useful to build a network that does space processing like the human brain does is it's relatively easy to give a computer system good spatial information and to make a good spatial model of the computer's environment using a range of sensors and cameras. So if you have cameras all around the room, you can make a very effective model of the space within the room without too much trouble. So that's not a difficult computer vision problem. So spatial cognition is not, therefore, an obvious target for deep learning in computer vision. Though, indeed, one of the advantages, one of the things that works very well in Tesla's autopilot system is that it's primarily built from deep convolutional neural networks doing spatial processing from a limited set of cameras. Okay. For humans, we have to rely on a very limited set of cameras. We have the inputs from the two eyes, and this shows how things change over time, and we can analyze that to give an internal three-dimensional model of our environment. Okay. So these two pathways are the pathway for sort of object recognition and for spatial processing are really quite separate, and damage to them produces very, very different deficits in perception. So here's one.
I can't tell. Can you see it? I can see it, but I can't tell. I can't even reach it.
You seem to do better with your left hand. And take a crack at it if I hold it up closer. All right, how about now that you've touched it with that hand? Can you reach with the other hand and grab it? With your right hand?
My right hand.
So he has a really severe spatial cognition deficit as a result of a brain injury. And so that is, it doesn't really matter what the object is here. He's just not going to be able to interact with it. Not going to be able to sort of find his way around the space and find it in space to perform actions. All right. Damage to the ventral lobe has a very, very different effect, known as visual agnosia, which damages people's ability to recognize objects. So let's see what that looks like.
Let me give you a hint. I'm going to give you a choice, and you tell me what this is.
Okay, I can tell you then.
Okay. Is this a clock, a lock, or a telephone?
A lock.
Is it a lock?
A clock, a lock, or a telephone? Okay, let's rule out a telephone. A clock or a lock? Clock. Gotta be a lock. Must be a lock then.
Are you sure?
No, I'm not. But I don't recognize it as a telephone.
Mm-hmm. Do you recognize it as a lock?
No, honestly, I don't. But it's just it really looks very indescript to me.
Uh-huh. But your hands know what they want to do.
I know,
with
and
it.
they want to do something.
That's right.
I can see them.
Uh-huh.
Yeah, I can just picture them, and I know that I have. It's like I'm undoing a combination lock.
Good.
Huh.
Okay, good. We had to work on that one, didn't we?
No shit.
That's okay.
There's a couple of things to notice about that, right? First of all, when he was even having trouble keeping in mind the concept of these different things, right? She'd say the words to him and be like, I'm going to have to repeat those to myself. I can't even keep in mind the concept of these objects, which is a really intense deficit that he's having. The second thing that you may have noticed is that he was trying to figure out how he'd work with it with his hands, right? And one of the main roles of the other visual stream, the dorsal stream, which he doesn't have damage to, is to plan actions, right? And he seems perfectly able to plan actions with this thing, even though he can't name what it is, right? So it seems that even object recognition has like a motor planning component to it, and vice versa, right? And his motor planning component of object recognition seems quite intact, which is a very surprising thing to see there. Okay. So V1, as we've seen, is a visual field map with a spatial layout. Beyond V1, we have a series of further visual field maps, at least 30 of them over our different processing streams. All of these have a spatial layout. So if we look at, you can see a bit of V1 at the bottom here. You can see that the sort of polar angle here, so that is different directions in vision, are represented by different colours. And you can see at V1, you see this fade from green here, that is, say, the bottom of the visual field, at the top of V1, down to blue. And if you went further, you'd see it continues down to red, but we can't see that because the image gets cut off. We see the same in this area here, which is much, much further up the visual processing stream. This is area IPS 0. Again, we see this gradual fade from greens through blues, which is horizontal, and then to reds, which is vertical again. Okay. So all of these all have a sort of spatial layout, right? And we've seen that in deep convolutional neural networks as well. They maintain the spatial relationships up into the higher levels as well. Right. Now, we can think of these different areas as layers of a deep convolutional neural network, but the hierarchy of these areas is nowhere near as linear as in an artificial network. All right. So we've got a simple pathway coming up from the retinal ganglion cells to the lateral geniculate nucleus of the thalamus to V1 to V2. That's pretty linear, right? But beyond that, all right, as we go through more stages, we see this kind of split off to lots of different areas in different directions. Okay, so as we go through more processing stages, we see more and more areas, but we also see more and more pathways, more and more directions that the the lines are going in. Okay. So what we've seen in artificial networks is we have just step by step by step, right? If we were to think of this in the same way, we'd have another pathway going off here and another pathway going off here, all of them aiming to achieve different things, right? The difference here, so in the early visual cortex, we saw that there are populations of neurons achieving different things with different responses, but these are all held together so that their responses can be analysed together. When we get into these later areas, it doesn't look like that anymore. The responses are really separate, suggesting that actually they don't need to be analysed together. They don't contribute to the same task, right? So they contribute to really quite separate tasks here. Okay. So we've got these different areas that are in these sort of higher layers of the network that are each trained to achieve a different task, right? For example, in very coarse sense, as we just saw, object recognition and spatial processing, right? So we've got two completely separate networks involved in those two different tasks. An artificial deep convolutional neural network, as it's currently conceptualized, is trained to do one single task. Right? A human can do lots of tasks using the same visual input, right? And it would likely cause conflicts and problems if a single area had to be trained to do many tasks with conflicting demands from each other. All right. Here you can see these areas kind of color-coded to the early visual cortex in the occipital lobe, the dorsal stream up into the parietal lobe, and the ventral stream going down into the temporal lobe. Going up the hierarchy of the early visual areas from V1 to V2 to V3 to V4, which is the start of the object processing hierarchy, all of these areas have a larger representation of the central visual field. So low eccentricity here, small numbers on the x-axis mean the central visual field, so where we're looking, right? And we can see that here we have more millimeters of cortex per degree of retina, right? So per amount of visual space, right, we have more amount of visual cortex processing it. So that basically, as we discussed before, we have an over-representation of central vision. Okay. And that over-representation actually becomes greater for later areas, so particularly for object recognition areas. They really over-represent central vision. So if we look at area LO here in purple, its representation of peripheral vision is very small, so very few millimeters of cortex per degree of visual angle. But its representation of central vision is actually even larger than in the primary visual cortex. We also see that the area under these curves comes down. So if we look at LO1 and V4, they're much, much smaller than V1 and V2, which we see here. And this resembles something that we also saw in deep convolutional neural networks, that the early layers, right, there's not very many feature maps, but they're very, very large spatially. Okay? But as we go up, we get more and more feature maps, so we can see that as more and more layers. Okay? But they become smaller and smaller spatially, okay, like we see here. Okay. The different visual field maps here specialize in different things. All right? And so far we've seen this separation of form processing and motion processing. But that is actually, space processing as I've described it. That's a relatively big distinction. But actually beyond that sort of crude distinction between form and space processing, it's been very hard to understand what each of these 30 visual field maps is actually doing. Now, that's partly because humans like to think of an area of the brain having a specific function, right? And indeed they do, but what they are is they're layers of neural networks. And we're very bad at thinking of the function of one of these areas as being it's a layer in a neural network, right? So the function of each of these layers is performing a particular feature transformation, as we've seen in later areas. Humans are really bad about thinking in terms of deep network feature transformations, but the brain is very good at doing them, right? And computer processors are very, very good at doing them. My high school biology teacher used to say to me, if the brain was simple enough for us to understand, we'd be too simple to understand it. Because the only reason that we can think in complicated ways is because we have such a complex brain, right? But we're very bad at thinking about how it works. It just doesn't come naturally to us because it's not the type of problem that we normally need to solve. Okay. Indeed, because we haven't got a module that does it. We've never been trained to do it. We haven't spent our lives thinking about these things. And if you do spend your life thinking about these things, they become easier to understand and become better at them. Okay. Through these hierarchical stages, right, in these multiple networks branching off in separate ways, we're just going to look at them linearly, but remember that they do tend to branch off in complicated ways. Through these hierarchical stages, we find increasingly complex feature representations, right, over increasingly large areas of visual space. All right. These spatial and feature transformations are occurring together, right? The integrating filters span both the spatial representation and also a range of features, just like the color filters that we saw in the retina. Okay. But the feature transformations are much more complicated than this, as we saw in the last class. We do have representations of lines and edges, but we don't have representations of shapes and objects leading to faces. What's going on in the middle is neural network transformations instead. Okay. The spatial integration is actually much simpler. So we're going to look at this first. All right. So as we already saw, receptive fields get larger going from the center of the visual field, so that's where we're looking, to the peripheral visual field, meaning that we have a finer representation of space in the central visual field. Okay. As we move from early areas, V1 is at the bottom here, to kind of middle level areas in shape processing, so LO1 here. We see that the receptive fields get larger and larger, that is to say, more and more visual space is being integrated into the neural response in each of these layers. Okay. The neurons are responding to larger areas of visual space, following what we might think of as repeated convolutions with certain spatial extents. Right? So kind of more integrating more and more information. Later areas in this network are no longer sampling from the retina or from the visual image, so as I say, it's good to get away from that idea. Their input is often coming through V1, right? Or indeed their input is always coming through V1, and they often are directly connected to V1. Okay. So because a higher layer in this network is inheriting much of its receptive field size from the earlier layers in the network, right? So these neurons in V1 have a particular spatial area that they'll respond to. So later areas sampling from them might extend that spatial area, but a lot of it is just going to become because it's going to be they respond to the areas that the earlier that their inputs respond to. Okay. So because a higher layer is therefore inheriting much of its receptive field size from the receptive field size of its inputs, any questions about spatial integration need to take that into account, right? That what the integration is not the size of the receptive field here. What V2 is doing is the difference in size there. Okay, so that's what V2 is adding, is that small difference there. So we can actually look at the spatial extent of pooling between different visual areas using a couple of different techniques that we're going to look at here. Okay. So first of all, you can get this, these receptive field sizes that we see in all of these areas here. And because they all are taking their inputs through V1, we can first of all subtract V1's receptive field size to say, well, how much is their sampling extent from V1 in terms of degrees of visual angle, right? So how much extra visual space are they adding to their response in each of these layers? Okay? So V1 isn't included here because this is all relative to V1. Okay. And if we think of these as sampling from V1, well, we then have a V1 sampling extent represented in degrees of visual space. And as we saw earlier, we know this thing cortical magnification factor, which gives us the amount of cortical surface in millimeters per degree of visual space. Right? So that actually lets us take these numbers here, this sampling extent in degrees, and then convert it into millimeters, because we know how many millimeters are in each degree. Okay. And when we do that, so what we're basically doing is multiplying this visual space sampling extent by the spatial representation in V1. What that does is it converts the sampling extent into millimeters of cortex. All right? So this is how much of V1 V2 is integrating the responses from. Okay. And actually what we see here then is that this integration kernel that we see here, which might be considered something very much like a convolutional filter, is first of all, it's constant over the whole visual image, right? So that tells us that regardless of where we are in V1 and V2, right, V2 is always sampling from a constant spatial extent of V1, right, much like we would expect in a convolutional filter. Right? So even though the weights aren't shared between the filters at different positions in this image, right, the spatial extent of the filter in terms of V1's input is shared. And indeed, when we go up to later areas, we see the same sort of thing. Okay, this 2 millimeter sampling, it's about 2.5 millimeters that we see here, very closely resembles the size of a hypercolumn that we saw earlier. Okay, so that hypercolumn is the bit of cortex that represents one spatial position and all of the features within that. So it seems that V2's inputs are sampling across all of those feature maps, right, regardless of where you are in the visual image. So in a biological deep network, a feature map may be biased towards the center of the visual field, the bit that's most useful to us, right? But within that representation, there does appear to be a common spatial extent of sampling to the next layer. Okay. There's another method that looks at, this is kind of a mathsy way of deriving how this might work, right? There's another method of doing this that works perfectly well as well. What you can do is you can take the response of a recording site in V2, in this case an MRI voxel, okay, and you can attempt to predict this response using the sum of the responses of a group of neurons in V1. That is to say, if this group of locations had their responses summed together, that is to say integrated, they might produce a response that looks like this. Okay. So what we do then is we take all of the responses of the recording sites within this area here, right, which is what we see here, the V1 signals, and we add them all together, and this gives us a prediction of what we might expect to see from a neural population that was integrating all of these responses. Right? And then we can move around this, this kind of sampling kernel and try lots of different positions and lots of different sizes. Okay? And we can see which which prediction best fits the data that we have here. So basically we're fitting the position and the size of this sampling kernel. Okay. When we do that, very much like we just saw, we see that V2 is sampling from a constant extent of V1. Very much like here, it's around 2 millimeters, 3 millimeters. Okay. V3, which we see in yellow here and green here, seems to be sampling from a very consistent extent of V1, where it gets most of its input from as well, though it's a little bit larger than the extent from V2. And V4, again, seems to be doing about the same thing. So it seems to be, again, integrating about the same amount of spatial information from V1's representation. Okay. So these two different methods work in very different ways. One's basically maths, and the other is just sort of throwing stats at it. Yeah. And they produce a very, very similar response, which is consistent with the idea that that later areas in biological neural networks sample from fixed extents of earlier areas. So just like we see the convolutional filter is normally a fixed size in artificial networks, the brain actually does seem to be doing something quite similar. In the brain, that may result from there's just only so big that a dendritic tree can grow. All right? And that may be the limit there. A complicating factor in all of this is that the cortical sampling extent is affected by what we're doing. Yeah, so, okay, sorry, just to sum up that. This brings up something that I mentioned earlier. A receptive field in a biological system is the sampling extent of the visual image. All right? So if we have a neuron in anywhere in the brain, how much of the visual image does it respond to? That's a receptive field. This sampling extent between visual areas is often referred to as a connective field. Okay, so that's the sampling extent of the previous visual field map. So this will be in millimeters, whereas the receptive field size will always be in degrees of visual angle. A complication to this that becomes very important in our next class is that the size of this sampling extent depends on what we're doing, right? In a artificial deep convolutional neural network, The sampling extent is fixed and is not affected by what the what image is shown. Right? Here, though, we've shown that if you show people visual images, the sampling extent is larger, and if you show them nothing, the sampling extent is smaller. Okay? So there seems to be a kind of dynamic allocation of the sampling extent depending on what we're doing on things like attention. So we'll come to that a lot in the next class. So the brain seems to reallocate resources to fit the context of what it's doing, which artificial neural networks cannot yet do. Okay, so we've looked at a few things about spatial integration through the visual hierarchy. Right? We saw that visual cortical areas beyond V1 also map spatial, the visual field spatially onto the cortical surface. Right? We saw, therefore, that spatial relationships between image locations are maintained in these later visual field maps as well. We saw that there are multiple branching hierarchies of the visual field maps, largely because these are probably performing different tasks, these different branches. They're kind of going off to perform different tasks without interfering with each other. The main division between these sort of two different branches, the first two branches that you see, is between the ventral and the dorsal streams, which are focused in the temporal and the parietal lobes, respectively. And these are involved in object recognition, which we're going to look at in some detail, and spatial perception and action planning, respectively. Okay. Finally, visual field maps throughout this processing hierarchy remain biased towards central vision, but later visual field maps seem to sample from approximately constant cortical areas of early visual field maps, regardless of which visual position they're processing. Okay. So the spatial integration story is pretty well pinned down within early visual areas, at least. The transformation of features, on the other hand, is difficult. It's really complicated, and we get to a point where we stop being able to think about it, which is fun. We can look at that now. Okay, so the other aspect of sampling filters is how they transform feature representations from simple features like edges that we see here, right, to higher level features that reveal the object's identity, like this face. Okay. So this is the pattern within a filter rather than the spatial extent of that filter. So how we get from this simple representation of oriented edges to a holistic expression of a face is a really difficult problem. Okay. And here we see a face that's been broken down into its constituent edges, right? And using these, we can still see the face. Okay. But the artist who made these pictures, this guy, Chuck Close, he has a neurological problem that prevents him from recognizing faces. This is a problem called prosopagnosia. And about 2% of the population have this problem of recognizing faces. And as his paintings show, the way he recognizes people is he relies on these simpler features. He looks at the simpler features to determine what he's looking at, and that can be very diagnostic. Prosopagnosia is a really funny condition. I have a friend with it, and he's American, and I see him at conferences every 6 months or so, and he recognizes me as being the guy with a beard and an English accent. That's pretty effective for him. He knows who's talking to him immediately. He does not recognize me by looking at his face. Oliver Sacks, who's a famous neurologist who recently died, he had it very, very severely towards the end of his life. And he describes the situation of looking at a bearded man staring at him in a restaurant window and not realizing that it was his own reflection. So it can be very extreme problem. But face processing is something that most humans are exceptionally good at. Okay, so when we want to go from this early sort of edge-based representation up into a representation of what an object is, the first important step is to group together edges that might have something to do with each other, that might have some relationship to each other. Okay. So here we can see some possible groups. Here we can see edges that kind of feed into the next edge here, so this line here, you can see a few of those. And so this is often called a snake path. These edges probably belong to the same object as we would see this in the real world, because one edge kind of points towards the next one in space. Right? Alternatively, you might have a structure that looks a bit more like this. Right? So this is another possible grouping of edges that we might learn to see. Okay, or you might have a structure that looks like this, where we're grouping together all of the edges around this circle so that we see a larger figure emerge, like a circle here. Okay. The ability to do this, to pick out groups of edges, right, is actually not present at birth. If you get young children and you show them displays like this and say, well, where is the group of edges, right, they just can't do it. Their performance is extremely poor. Okay. And perhaps so this is what you see here. Even at 5 years old, children are really bad at doing this, whereas they can tell you the orientation of the line perfectly well. Okay. If we look at even 13 and 14-year-olds, their performance at doing this, their ability to group together a bunch of edges, is considerably worse than we see in 20 to 30-year-olds. Okay. So this sensitivity to grouping together different edges seems to increase deep into adulthood, which is a really rare developmental progression. Okay. So we saw earlier that things like edge detectors may well be hardwired, right? There are really simple filters that might develop to do this. But it seems that beyond V1, when we start having to group together edges into larger scale structures, it seems that the structure of the filters has to be learned, and we continue learning this until quite late. And it seems to be not based on backpropagation of error, but instead depends on developing responses to patterns that we've seen before through Hebbian learning. Okay? And so this depends on, this development of this ability is likely to depend on learning the statistical structure in the world. That is to say, patterns that we've seen before. Right? And so it seems to take a long time to develop. And this seems to be one of the first filters that we need to do to perform an object recognition task, to group together edges. Okay. So this is the type of problem that we're faced with when we're trying to think about what type of later feature maps we might need to develop to solve this type of problem. And even beyond V1, it's even hard to figure out what V2 does that's different. Okay. Generally, V2, the next visual area, responds very similarly to V1 when presented with oriented edges or shapes. Okay. Now, because we've seen that V1 only responds to spatial frequencies and orientations of a black and white input like this. Okay. So we can make an image, like these images here, that match the distribution of spatial frequencies and orientations in these images on the left here. Right? So this is just a noise pattern, so it doesn't have much structure to it. But it does have the same pattern of spatial frequencies and orientations. Right? It just doesn't have any of the local correlations that we would normally expect to see here. So the image has basically just been scrambled up. Okay. And so here we can see kind of matched images for all of these. And actually, if we show these images to primary visual cortex, it responds just as well to images like this as it does to images like this, which is a real surprise, because we see images like this a lot, and we never see images like this. Okay. But if we look at V2, it does not respond well to images like this at all. Okay. To make an image, to make a response that looks like this response, you need to have an image that looks like this. So that is an image that looks much more similar. Okay. The way these images are constructed that drive V2 well, okay, is as well as having the same patterns of orientation and spatial frequency, the same amounts of each orientation and spatial frequency as these images here. We also need to have the same correlations between different orientations and spatial frequencies. Okay. So we can think of these operators here as kind of grouping together responses across different feature maps. Okay. So what we're doing is we're saying, well, our filter, right, this is basically being a filter, takes inputs from all of these units in the earlier layer, okay, and then that predicts how V2 will respond. So this is really neural network-like operation. So if you show images like this and this to V1 and V2, V1 responds just as well to this and this. V2 only responds to this. It doesn't respond well to this at all. Because the images that are normally present in natural images, right, so the structure that's normally present in natural images, correlations between orientation content at different locations, is not present here. So V1, which has learned everything, learned how to respond based on those patterns, doesn't respond. Okay? It needs something where those patterns are maintained. Okay. So this is a neural network answer to what V2 is doing. Beyond V2, we still don't have a good feeling for what drives the responses of visual areas, but we now understand that it's likely to be a feature transformation from the outputs of the previous layer. Okay? So if we look at V2's outputs and we do some computation on those, then we might be able to predict what V3 is doing. Okay? So basically, if we were to perform this same operation again on the set of units that emerge here, right, we might be able to predict what the next layer is doing. It becomes very, very difficult for experimenters to think about neural representations and transformations in the next layer after this. Because what we're doing here is, look, we've got a pattern of responses. Okay, so here we've got an image. Here we've got a pattern of responses to that image. Here we've got some pattern of responses in the pattern of responses to that image. Right? The next layer would be looking for a pattern of responses in the pattern of responses to the pattern of responses to that image, which is a very, very difficult thing to start thinking about. Right? So you're looking for a pattern within a pattern within a pattern. So humans are really terrible at doing this. Okay? And it becomes very difficult for us to think about these transformations. Okay? So if we want to make a hypothesis about what later levels are doing computationally, the answer is likely to be in deep convolutional neural network terms. Right? Like this is an answer in deep convolutional neural network terms. Okay? So deep networks then become a very useful tool for guiding the hypothesis and understanding what we might be looking at, what we might expect to see in later areas after this. Right? Which is likely to give a more accurate result than what we started by talking about, things like, oh yeah, where you've got conjunction of two edges, right? That isn't actually what you see. But something like where you see a pattern within a pattern within a pattern, that might be much more like it. Okay. So the layers after this become really difficult to think about, but something interesting happens after a few of them, once you get into the inferior temporal cortex, which is this.
They can record from single neurons in part of the inferior temporal lobe, and these neurons will be activated by faces better than by any other kind of stimulus that they can present. Not only will they be activated by faces, but if the faces are distorted in certain ways, the neurons will not respond.
As we watch, we can hear single cells in the inferior temporal lobe of monkeys reacting to a single monkey face. The cell only fires if the face is at the right angle and is not distorted.
Okay, so that's an example of an object-selective neuron, a neuron that responds to a particular type of object. In this case, face is used as an example. Faces are a common model for object processing, partly because humans are very social animals and care a lot about faces. These recordings are taken from a monkey, which is why you're looking at monkey faces there. If we look in the human brain using functional MRI, we see areas that respond more to faces than to other types of objects. So if we look at all of these 6 examples here, we see the same blob lighting up as responding more strongly to faces than other objects, than scrambled faces, than houses, than hands, and so on. So it shows that the brain also contains similar neurons that respond to faces. But looking at a whole brain area like this, we'll group the responses to all faces. Within these areas, within this area in humans, there are even cells that respond more strongly to specific faces, importantly regardless of the image used and therefore regardless of the activation of the retina. So here we see a neuron that was recorded in a human in the fusiform face area during open brain surgery to treat epilepsy, which is a particularly excellent way of getting types of recordings like this, because otherwise it's very hard to make holes in the human brain to do neuroscience with. So this neuron responds very strongly to Jennifer Aniston, regardless of the angle and size and position that she is in the image. It responds less well to images of Julia Roberts, which is some of these images on the bottom here, even when Julia Roberts is presented at a similar angle, size and position as in the pictures of Jennifer Aniston. So this shows us here that the response is not driven by what the image looks like, rather is driven by what the image contains, what type of object is in the image. But given the complex nature of feature map representations, perhaps we shouldn't expect a single neuron to respond so cleanly. And in fact, if we look at the last slide in detail, you see that there are some responses to other types of images as well, just the strongest response to Jennifer Aniston. Right? So rather than seeing that neuron 1 responds to Bill, neuron 2 to Samantha, neuron 3 to Roger, we might see that neuron 1 responds more to Bill and less to Samantha and less to Roger, but still does respond a bit to these other ones. And the other neurons do respond somewhat to Bill as well, though not as strongly as neuron 1 does. Okay. This is known as a distributed encoding system. So here, the pattern of activity across a larger group of neurons is used to represent the identity of the person. Okay, so this is what the middle layers might be doing. Then the object's identity isn't straightforwardly reflected in the activity of a single neuron, but rather in the pattern of activity of a larger population of neurons. This has some advantages and disadvantages. The main advantage is that it allows some of the cells to die without this representation failing. So this is known as graceful degradation. And so, for example, if the neuron that responds to Bill dies, if it's the only neuron that responds to Bill, and it responds to Bill and nothing else, right, then the subject, the person, won't be able to recognize Bill anymore. But in a distributed encoding system where the other neurons are responding somewhat to Bill, most of the pattern will remain after one of the cells dies. Okay. Second, this allows new patterns to be stored without making new cells. So if we've got this fixed population of cells like we do in the adult brain, new objects can still be stored within that population. So then a fixed group of cells can store a variable number of neurons, which is particularly useful as we move around the world and see new types of objects. Okay. The final advantage is that this is consistent with measured cell responses. Cell responses are rarely all or nothing like we saw with the Jennifer Aniston response. Most of the responses actually are much more graded and will respond a little bit to one thing and a little bit more to another thing. Okay. So this really fits with how the data actually is. The disadvantage of this is that it's really hard for humans to understand. An all or nothing type of encoding, where one neuron responds to one object, is really easy to understand, but a distributed pattern is harder for us to get our head around. But also when we make recordings from those neurons, it's harder to see the distinction between the responses to different inputs, because we're only looking at the response of one neuron. If we can take a measurement from all of the neurons, then that would be just as easy to understand, potentially, particularly by analysing that response a little. But it becomes harder to make recordings from lots and lots of neurons simultaneously, just in practical terms. Okay. An important thing to note about this distributed representation is that the last layer of an artificial deep convolutional neural network for object recognition is fully connected. So this last layer can allow a decision based on the pattern of responses across all of its inputs in the previous layer. Okay. So in a computation like this, a distributed pattern will allow the right decision to be made just as easily as the response of a single cell will allow that decision to be made, because the fully connected last layer can look at the pattern across all of the neurons before making the decision. So although we don't have a good feeling for what's happening in the middle layers between V2, which is looking at correlations, and these object selective areas, we believe that they're using deep network structure to transform the visual image representations to object representations. if we compare the brain's object recognition network to a deep network, we can see that there are many similarities in the spatial representation of features and in the spatial sampling between layers, as we've seen throughout this lecture. We've seen that early layers are doing simple edge detection, and that layers, once we get to V2, seem to be just forming associations between the output of earlier layers. Okay, so finding patterns based on experience of correlated response patterns that they've seen before. And of course, it's again important to remember that these later layers are not receiving image inputs; they're receiving inputs that that are a pattern of neural activity in a previous layer. So we understand the processing step that's occurring at each level, which is just drawing associations in the activity of previous levels, previous layers, but it's hard to intuitively understand how this will work over many layers, which may be why it's taken so long for us to understand. Effectively, what these later layers are doing are looking for patterns within the patterns within the patterns within the patterns. That's hard for us to do, but it's relatively easy for a computer to do, and it's also very easy for neurons to do. So we can simulate this process and see how the simulation performs to see whether this is indeed consistent with the responses that we see. Okay, so responses of later network layers, like we see in red here, closely resemble the responses of face-selective cells, shown in black here, a cell from the inferior temporal cortex of the macaque. So this is our current best theory for how object-selective responses arise, is that there's lots of middle layers that are doing relatively simple computations. It's just hard for us to follow those computations through. Importantly, we see very similar responses to faces, even if the images vary greatly in position, size, and viewing angle, like these faces do in these images here. If we look, though, at image sets with low variation in position, size, and viewing angle, so where the face is always presented quite similarly, or the object is always presented quite similarly, okay, then many simple computational models have a high object recognition performance that is comparable to what a human can do in classifying objects. So V1-like models and V2-like models do quite well here. Okay. But once we introduce more variation in position, size, and viewing angle, then the population of IT neurons does much better than the population of any population before it. So it does much better than the population of V4 neurons. And a deep network here, HMO network for hierarchical modular organization, does much better than the simple neurons the simple models that we see in grey. Indeed, humans, IT neurons, and inferior temporal cortex neurons, that is, and deep network models all do very well here, while simpler models all do very poorly in understanding the image, recognizing the object in a high variation situation. Okay, so to summarize the last part of the lecture, okay, we've now seen that the ability to group edges into more complex patterns continues to develop into adulthood, which suggests it relies on learning, although earlier edge detectors that we've seen right at the start of this lecture may actually be hardwired and may not rely on learning at all. It seems that these later layers and middle layers basically do transformations that rely on finding commonly seen patterns or patterns that have been seen before in the activity of earlier layers. Okay? And these have traditionally been very difficult for human experimenters to understand because we like to think in terms of the image rather than the pattern of activity in the earlier layer, which has always been a very difficult thing to measure anyway. Okay. We've also seen that later stages are likely doing a very, very similar computation to what we saw in V2, but from more abstracted inputs, so from the outputs of earlier transformations. Now, it's hard for humans to think about these representations and transformations, these patterns within the patterns within the patterns, right? But we can simulate them relatively easily using artificial deep convolutional neural networks. Later in the ventral stream, we find face-selective neurons, And it has traditionally been very hard for us to determine how those are arising. But if we use artificial deep convolutional neural network simulations and train them to do object recognition tasks, then they do end up with face-selective responses very much like the ones we see in the inferior temporal neurons of monkeys and of humans. Okay, so at this point, we've looked through early visual processing enough that you should find the first of the papers from our reading list relatively easy to follow through. This looks at the representation of visual information in the early visual cortex, so spatial information and orientation information like we saw at the start of this lecture. Okay, so see you on Friday at 9:15.